So we're talking about how to sequence DNA. And the way this works is through a DNA polymerase, taking whatever template of DNA that you're trying to look at, and the DNA polymerase makes a copy, and you throw in these dideoxynucleotides. So the individual bases that make up DNA, these dideoxy ones will like poison the reaction. And so as you're making a copy of the DNA you're trying to sequence, at any one of the bases it will stop because this little nucleotide poisons the reaction. And what you generate is randomly sized fragments that represent what the sequence was that you were making. So the first sequence in this reaction started and then ended with a T. And so I have a very small piece that ends in a T. And we were talking about how these are fluorescently labeled. So each one of these nucleotides that stops the reaction fluoresce a different color. So this is a cartoon of what the imaging, the sequencing machine is actually looking at. But what it's saying is the smallest piece that went by had a red dye on it. So that red dye indicates that it ended with a T. So I know that that base is a T. And then one base pair away, the machine recognized that a yellow fluorescent molecule went by. So it was one nucleotide longer, it's yellow, and that means it was a G. And so as you read up a gel, you can read the actual sequence of what the enzyme was making a copy of your original. So you're reading a copy of the original. So I read this gel as TGC, GGG, CTT, AT, and so on. And I'm actually sequencing the DNA. So this is the traditional dideoxy Sanger method that we talked about last time. And we started talking about the next generation sequencing technologies. And Sanger sequencing is robust. It gives you a really fairly high fidelity readout of what the sequence is. That is, it makes mistakes very rarely. And so you're pretty confident about what your sequence is. The problem is it's expensive. DNA polymerase is expensive to isolate, and making you can only go, and it can only resolve for as long as your gel is. So you end up having to do short fragments of sequence, and then you have to shift over and sequence the next fragment and then shift over. So that's time consuming every time you're using expensive enzymes. And so the next generation sequencing are trying to take advantage of trying to increase the output flow, increase how fast you can do it, and also decrease the cost. So we talked about pyrosequencing last time, and I'm going to show that video again, and we'll just talk about pyrosequencing once again, and then we'll talk about hybridization sequencing. All right, so pyrosequencing. Well, let's just watch the movie here, and then I'll talk about it. Okay, so here's the template that we're trying to sequence. And in the solution is DNA polymerase. So DNA polymerase is the enzyme, and that's the functional thing that's actually copying and allowing us to read the sequence. Okay, we're not looking at the sequence directly. We're looking at what DNA polymerase is looking at, and we're looking at a side effects, secondary processes of what it's done. So it's moving along. This is the template strand, and DNA is double-stranded, so the enzyme is coming along. It recognizes this nucleotide as a C, and so it's going to incorporate a G nucleotide in here. All the nucleotides that are going in are nucleotide triphosphates, so ATP or TTP or GTP. And when they actually get polymerized and put into this new strand that the enzyme is making, it cleaves off two of the three phosphate groups. And so the two bases in this newly formed piece of DNA is just connected by one of the phosphates. So two phosphates are going to come off. In pyrosequencing, what we're doing is watching how many nucleotides got put in. Okay? So in this first step, I've put in all G nucleotides. So I've got my template, I've got my enzyme in a tiny little compartment. And then I just add in G nucleotides, and I see what happens. Okay? So what happens is that G nucleotide got incorporated, and the two phosphates came out. I went from a triphosphate, I cleaved off the two phosphates, and those ones come out. that enzyme took those two phosphates and created an ATP molecule. Okay? So what I've done is I've detected that a single G has gone in because two phosphates came off, and what was produced then is a single ATP molecule that has come out of that. There's a second enzyme in there called luciferase. And luciferase is going to take that ATP molecule that we just generated, and it breaks it, and when it does that, it exposes it to light, or light comes out of the reaction. So what I've done is I've detected that a single G went in, because all I gave the reaction were Gs. That's the only nucleotide I allowed anything to happen. There was a single C in there, so a single G got incorporated, and what the computer reads is what was the intensity of the light that came out. Okay? The intensity of the light came out appropriate to one G being incorporated there. So I got this little peak. Then once I see a peak, we clear away all of the nucleotides and you add another nucleotide. So I surveyed for G's presence, there was one G. Now I'm going to move on, I'm going to survey for the next. So the enzyme slides along, and then I give it new nucleotides. In this case, I've given it A's. 3 A's are going to get incorporated. 3 diphosphates come out. 3 ATPs are made. And my luciferase converts those 3 ATPs into light, and the computer detects the presence of 3 nucleotides went in there. So now I've sequenced. I've said I know it's GAAA is the sequence so far. So I've surveyed to see if there are any A's present. Now I wash away all the A's, and I add another nucleotide. Yeah?
Do the peaks just correspond to the amount and not the specific?
It's just the intensity of light.
Okay.
The reason I know it was A's is because that's all I've given at this step.
Okay.
Right? So I now give it more nucleotides. Here I'm giving it T nucleotides. Now these are C's. I'm sorry, these are G's. So the T's are not going to react at all. Right? So I got nothing. Nothing when I added T's. So, okay, nothing was there. You just move on and try the next base. Now I've thrown in C's. 2 C's are going to get incorporated, generate 2 diphosphates. Those 2 diphosphates can turn into 2 ATPs. And so I get a double peak of light intensity that came off of 2. That's a bummer. So this is pyrosequencing. You're watching the sequencing. The reason this is quick and efficient, it seems tedious because you have to keep putting single nucleotides in one at a time. But you don't have to sequence it by running this out on a gel. You're watching in real time those nucleotides come off. So. Do you have a question?
Well, when you were saying, so you do G, and then you washed away G, you screen for A, you washed away A, you screen for C.
Yeah.
Because the sequence can be fairly like random, you could have had GA, 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 or something
yeah.
to that effect. It's time taxing because you are trying to screen for T's and C's, and really it's...
Yeah.
So it just takes more time to screen for each one.
Exactly. If your sequence was TA, 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 to know that you would have to put in T's, see T's get incorporated. Yeah. And then you'd try G's, oh, no G's. Right. Try C's, oh, no G's. So you'd have to, every time you do it, you have to survey for all 4 nucleotides. Then you move over and you get some peaks, or you won't get any peaks, and then you move to the next one. So every time you have to keep flowing through. So. These enzymes go really, really quick. So even though we've slowed it down here, this is really high throughput. We're just washing nucleotides by just as fast as we can, and they're just flashing light. Boom, 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 boom. And you flash. So it's the limitation of this machine is just how fast can you get a new nucleotide in there. The flashes are going to happen virtually instantaneously, and then we just wash it away, get the new nucleotide in there as quick as possible. This actually is really, really quick. Much quicker than waiting for a whole Sanger sequencing reaction to go, running it out on a gel, letting the machine detect all the fragments. That actually takes much more time than the high throughput flow machines that we can get. We can flow solution past these really, really quick. So, yeah. Questions about pyrosequencing? All right. If you thought that one was challenging, we're going to talk about solid sequencing. And actually, let me go back here. This is one of the examples of hybridization sequencing. Okay, so pyrosequencing, like Sanger sequencing, it's relying on a polymerase to do the work for you. Okay, in Sanger sequencing, the polymerase was putting in nucleotides, and we stopped the reaction whenever we put those dideoxys in. In pyrosequencing, we were also using a polymerase, but we got to watch it in real time at least. In hybridization sequencing, we're not even going to use a polymerase. We're going to use specific base pairing to tell me what the sequence was. Instead of an enzyme moving along and putting the nucleotides there and the enzyme reading the sequence, we're going to stick in fragments, and whether or not they're capable of base pairing is going to tell us what the sequence is. Okay? So this is sequencing by oligonucleotide ligation and detection. So instead of synthesizing a new strand, we're just going to put in a bunch of small nucleotide oligos, short little pieces of DNA, see if they hybridize, we'll ligate them together, and there's a detection method. All right, at this point, I'm going to turn on the volume here, and we'll listen to the woman describe this here. All right.
From the industry leader in DNA sequencing comes the next generation in genomics, the solid system, a revolutionary new platform with built-in scalability that delivers ultra-high throughput with unmatched accuracy. The robustness and flexibility of the solid system enables a wide variety of applications, including whole genome sequencing, targeted resequencing, gene expression, small RNA analysis, and chromatin immunoprecipitation. The workflow for each application differs only in the upfront sample prep and downstream data analysis. The sequencing chemistry and imaging steps remain constant. The solid system allows researchers two choices for sample preparation: fragment library or a MatePed library. In both cases, target DNA is sheared to a specific size, and adapters are ligated to the ends of the resulting fragments. A fragment library incorporates a single piece of DNA, whereas a MatePed library incorporates two DNA sequences that are a known distance apart in the target sample. The resulting libraries contain millions of unique molecules, representing the entire target sequence. Each molecule is then clonally amplified onto beads in an emulsion PCR reaction. The sample is...
Okay, so what they're doing is they're taking all of the sequence that you want to read, and they're fragmenting it into tiny little pieces. Okay, and so what we're showing here is these are just one of the tiny little fragments that you've generated from your original sample. Okay, so we took these tiny fragments, we stuck those little colored pieces that they showed you, little fragments on the end, and what they're doing is attaching those fragments to a little bead. This is a little silicon bead. So it's a very microscopic bead in a tiny microscopic water droplet. Okay, this is what they're saying is emulsion PCR. So what they've done is you've taken that fragment that you don't know what the sequence is, but you put fragments on the side that you do know what they are. Okay, you get that attached to a bead. So we covalently bond that fragment to the bead, and then you do PCR. PCR allows you to amplify a piece of DNA. You don't have to know the entire sequence of the piece. So since we put those ligated those fragments on the end, we can copy all of the pieces of that fragment. So on a single bead, you'll have thousands of copies of the fragment that you're trying to read. So we've just generically amplified it and got it stuck to this little bead. And this is all happening in tiny little water droplets. So instead of, we've done PCR in our labs, you actually set up the PCR reaction in that tiny little tube and put it in the thermocycler. In this, you've got just a single tube could have thousands of tiny little water droplets all emulsified into a lipid solution. So you have basically an oil solution in your tube, and in that oil solution is all these tiny little microscopic water droplets. In every single one of those microscopic water droplets is all these beads doing PCR reactions. This is going to now allow us to do what's called massively parallel sequencing. Because in our sample now, we have all these fragments, all of them are copied, and we're going to be able to sequence all of these fragments at the exact same time on this machine. Okay, so this is how you're sampling, you're prepping the sample. So I've got thousands of tiny little water droplets, each containing the fraction of that original sample that I want to sequence.
Is enriched for beads containing library template, yielding a greater than 90% positive bead count. Beads are covalently attached to a glass slide. The open slide format allows for the flexibility to analyze 1, 4, or 8 samples on a single slide.
Okay, so every tiny little dot here then is one of those fragments that came from your original sequence, that there's lots and lots of copies of every fragment there now. So what we have, and this is on a glass slide, like a microscope slide, and so you can have hundreds of thousands of these little pieces of DNA on that slide. And this technology is going to be able to allow us to sequence every single one of those fragments at the exact same time. So you can see how this is very, very quick then. I'm fragmenting, randomly making these samples, and I get to sequence all of those random fragments at the exact same time in one run. So Rachel.
Does the PCR reaction happen in each bead specifically, so then do you get a bunch of fragments
Yeah.
of that specific fragment?
Right, right. So in one tube, you can be doing hundreds of thousands of PCR reactions at the same time. They're all just happening in separate tiny little water droplets in there. So, yeah.
How do you know which part of the sequence they are? Like, how does it...
You don't.
Oh.
So you have to rely on a partial digestion so that you have overlapping fragments.
So then you just
Yeah.
do that.
So when you fragment, you don't fragment completely. You do partial digests so that you'll have overlapping sequences so you can put it back together. Yeah. Or you need to already know. If you're resequencing, you might already know. So for instance, the whole human genome has been sequenced. So if I were to sequence you, I don't have to create random overlaps. I could just sequence all the fragments in your DNA and then compare them to the human genome. I would know where all those fragments go now because I can use the genome that we have sequenced as a map. So if you're resequencing, you don't have to do it. But if you were using this to sequence a brand new organism, yeah, you'd have to do the same kind of conventional things of generating overlapping sequences so we know where they go. Okay.
With barcoding, one can pull up to 16 libraries, further increasing the throughput to 256 samples in a single run. On the solid analyzer, the templated beads are combined with a universal sequencing primer, ligase, and a large pool of dye-based probes. The dye-based probes are fluorescently labeled with 4 dyes. Each dye represents 4 of 16 possible dye nucleotide sequences.
Okay, so here's another thing that's going on. We're not going to actually be able to sequence on the first pass because we've got 4 probes in there. Each of the probes is specific for, well, it's got 2 base pairs in them, but those 2 base pairs could change, right? So on one probe, say for instance the one that's blue, might be AA and AT and AC and AG, right? So it's 2 specific or 2 specific nucleotides that determines how we labeled it. But on the first pass, we don't know which ones. We know the first position must have been an A, but we're not going to know what the second position was because there's 4 probes that all give the same color, okay? So this has to be repeated. This will become clear, I think, in a minute.
The complementary probe hybridizes to the template sequence and is ligated. After fluorescence is measured, the dye is cleaved off, leaving a free 5' phosphate group available for further reactions. This process is repeated.
Okay, so we've got, these are the small little oligos That's what the solid stands for, is oligoligation. So we've just created these small little pieces of DNA, and having these two nucleotides means it's got a yellow dye on it, okay? So the reason that's stuck is because those two are finding their complementary sequences here. The other things are just random fragments, right? They're all the possible sequences there. So this guy is labeled yellow because it's got two specific nucleotides there, but then there's thousands of other fragments that all have just slightly different nucleotides here, okay? So just think of that as generic. It'll just stick to anything. So what we're really reading is just those two nucleotides, because that's what we determined and what we actually labeled as yellow, okay? But that could be one of 4 possible nucleotide sequences, right? It's yellow, so it must be AT, or it could be AC, or AG, or AA, right? We don't know what that is yet. We just know that that first position must have been an A. We don't actually know what the second one is, okay? The next one that came in was labeled blue, and maybe that was GA, GT, GC, GG, right? So we know that that first nucleotide must have been a G. The way we're reading this is when the ligase comes in and ligates them together, it releases that dye and it's flashing as well. So it's kind of like pyrosequencing, but it's not a polymerase that's doing it. So every time one sits down and gets ligated in, it's going to flash a color, right?
For 7 cycles and yields a 35 base pair read. Additional cycles can be added to extend the read pair. The synthesized strand is removed. A new primer is hybridized, offset by 1 base, and the ligation cycles are repeated. This primer reset process is repeated for 5 rounds, providing a dual measurement of each base, separated by several cycles, which increases the sequencing accuracy.
Okay, so what we're doing is shifting over the length of where we start, right? So we started here, and these are the pieces that we hybridized in. Then you shift over a little bit, and you sequence again by doing the same method. So the first pass told you what the first base was. The other one was generic. The second pass, though, we just start 1 nucleotide later, and so the second pass tells you what the second nucleotide was, all the way along the thing. Then you extend 1 more nucleotide, and you do it all again. So you're sequencing each individual base at a time.
The solid system delivers the next generation today.
All right.
Build for scalability. The solid systems.
So I'm going to stop it here, but I want you to realize what's happening is you've got all these tiny little fragments, and what you're doing is just flowing over all of these possible possible oligos, short little pieces. And depending on which one incorporates, they're flashing a different color of light. So what we have is just all these on the plate. We're just flowing things across it, and there's a detector, basically just a big camera that's just taking pictures of this every time. So this little fragment, that tiny little dot right there, will flash blue and then yellow and then green and then blue and then yellow and then green. And so the detector is just detecting what are the flashes of light that came off of that dot. But since it's a camera and it's reading all of them at the same time, all of these things are flashing different colors of light the entire time, telling you what that first nucleotide was. So it goes through, everything determines what the first nucleotide was, then the next one, 8 base pairs away, it tells you what that nucleotide was, 8 base pairs away. So you get this like partial spotty sequence. I know this nucleotide, and then 8 nucleotides later, I know that one and this one. Well, then you just wash everything off, you put a new primer on that staggers you 1 more, and then you flow it over again. And then all of those dots flash, all the different colors. And the detector detects what all those little dots fluoresce that second time and analyzes it. And you do that 5 times, and then you've covered the entire length of every single one of those. You've got all those tiny little sequence fragments. Then you can piece them back together, and you know what the entire sequence was. So this is called massively parallel sequencing, right? Because I'm sequencing every single one of these all at the same time. And this ligation is really quick. I'm doing it in real time. I'm seeing exactly what got put in there. I don't have to like do a reaction, run it out on a gel and do all that. I'm in real time looking at all of these. So even though it seems cumbersome, and you're like, well, I kind of got one sequence or one nucleotide every 10 nucleotides, and I had to do it again and had to recalibrate. In fact, this is much quicker than Sanger because you're doing it in such massively parallel. Right? So even though it's kind of redundant in terms of what's going on in every single little spot, because it's all happening at the same time, this is actually really efficient. With this, we can sequence genomes in a couple of months now. A new genome comes out every so often, every couple of months. It took us years and years to do the human genome. And now we're like, oh, we did the rice one. Oh, we did the corn one. Oh, tomato just came out. Oh, whatever your organism that you're interested in, if you can get a grant for several $1 million, you can get the genome sequenced in a couple of months now. Yeah, Rachel.
Why is it 5 times that specific? Is it because we designed the fragments so they'll be...
Yeah, so the fragments that hybridize, and I can't really rewind here. The fragments that we hybridize, there's 2 specific nucleotides that determine what the color is, right? So it was a CA, CT, CG. Anything that starts with a C basically gets a yellow tag, for instance. Okay. But then to get that to hybridize, you have to have a longer piece, right? Because if you just had 2 nucleotides, they wouldn't efficiently bind to the template. So you basically take those 2 specific ones and then add it to another 8 or so nucleotides that are random. Okay? So you have to sequence kind of 8 nucleotides away from each other. So you're kind of leapfrogging. But then on the next round, you leapfrog 1 nucleotide away, and so you get the second one in all of those positions. And then you shift over 1 more and you leapfrog. So if you do that 5 times, you get a pass over every single one of them, and then you've reached the end of your fragment, right? So it's kind of redundant. You have to do it a couple of times to decide what the whole thing was. But the great thing is you get to do that redundantly over 256 samples every single time you do it, right? So, yeah.
It would seem that you're, in some way, I was just thinking like how they don't know the order of the genome already. Like you were saying that they have to be partial digest so they can put it back together if it's an unknown sequence.
Yeah.
But isn't it in some way breaking apart? Oh, you're saying it breaks apart the fragments in unknown places? Like, I would just think that because they're doing it in an order that the computer should be able to tell where it
goes back together?
Yeah, I just thought that.
Yeah, if you're resequencing, all right, if you already know what the sequence is in one individual and you're resequencing, then it will be able to find where all those pieces go, and you wouldn't have to have overlaps. But if you're sequencing a new organism's genome, you would have to go through all the traditional steps of like creating a random partial digest, cloning all of those partials into bacterial artificial chromosomes. But then when you actually have the fragments generated, getting the sequence is really, really quick now, right? Before we had to like get those big sequences, subclone them, do all of them by Sanger methods, 1,000 base pairs at a time. Here you can do hundreds of thousands of base pairs in one run. So actually the time of getting at the actual nucleotide sequence is trivial now. You still have to arrange it and get it all put back together in the chromosomes, and that still takes a little while, but actually getting the nucleotide sequence takes very little time now. So, yeah.
I have a question regarding the primer that we started with. Are they already primers that was designed? They are designed to have...
Right, we took our random fragment, and then we put the linker sequences on them. We know what the sequence of those linkers are. So that's where we make our primer, is in the known linker sequence. And so you sequence a little bit of stuff that you know, and then you get into the unknown. And then you make a little primer that's a little bit bigger, and we sequence some of the stuff we already know, but staggered over 1 base pair. So it's just a way to kind of initiate it. Once you get it initiated, it's going to go, and then it's going to sequence through your unknown stuff. So, okay, don't worry about too much about like how far it's staggered and what the dyes are. I just want you to see that number 1, you're doing it massively parallel, right? You get to fragment your piece, and then you get to sequence all of those fragments at the exact same time. And then also, this doesn't require DNA polymerase. This just requires DNA ligase. And so you're detecting the sequence in real time by hybridization, right, base pairing. And then the way you see the fluorescent molecules, because when the DNA ligase sews the pieces back together, you see the actual fluorescing molecule on the end. Okay? So it's hybridization rather than polymerization, massively parallel, and you don't have to have a DNA polymerase. Now, this is pyrosequencing, solid system. It's just like two of half a dozen clever little ways to sequence. I don't want to spend more time on that because these are the two main classes. One is pyrosequencing, the other is hybridization sequencing. All of the other methods fall into those kind of two general categories with just slight variations on these. And every company's got their own proprietary system, and they're trying to compete for how much it costs and how quickly they can do it. Since these prices are coming down, we're actually entering what some people call the post-genomic era and entering into the personal genomic era. Because if it's really quick to sequence, and if we can get the price down, we could start thinking about actually individually sequencing everybody. So right now, there's a couple of human genomes that have been done, but we're moving into if it's really cheap, we could just have everybody sequence their own genome, and we would know all of the alleles that everybody's carrying. We're not quite there yet. The sequencing costs are still prohibitive, but what we can do is what's called genotyping. Genotyping is a little bit more of a generic type of evaluation of your genome. It basically is a survey of all of the different alleles that you have, right? So instead of actually saying what is your actual nucleotide sequence, we're saying what normal known variants do you possess, okay? So genotyping is just any way by which you determine the alleles at one or more loci, at one or more locations, right? So if I want to know if I'm carrying a recessive allele for, cystic fibrosis. There's known alleles, known versions of the cystic fibrosis gene that cause the disease. So I could go be genotyped for that, right? They would just take some of my DNA and they would survey kind of generically what are the alleles that I have without necessarily sequencing every single nucleotide, right? So you can do this by multiple different ways. You can amplify by PCR. You could actually sequence. So in a way, sequencing is the most specific kind of genotyping you could do, right? To know what alleles you have, you could actually sequence every single one of your nucleotides, and we could tell, right? But there's more generic ways to do this. We could look for known polymorphisms, single nucleotide polymorphisms. So if we know that an allele that causes a disease is because you've got a single nucleotide changed, well, we could just go look at that single nucleotide. Don't have to worry about sequencing the entire gene. We just go to that location in the gene, sequence, and just look for that single nucleotide polymorphism. Or we could do AFLP analysis, fragment length analysis. Maybe one of the alleles fragments in a different way, so we could tell the difference between them just by doing a restriction digest. Or you could actually look at known repeat sequences as well. So I'll go through a couple of these and show you how some common ways they're used, but I just want you to get the idea. Genotyping is not sequencing, but it is determining what alleles you have. Okay, so not necessarily at the nucleotide level, but having some type of handle on what the allele is. So let's talk about microsatellite typing. We've talked about microsatellites before in terms of mapping, but you can use microsatellites for typing. So here is a little microsatellite region. Microsatellite regions are repetitive regions in your DNA, and they repeat for just a couple of base pairs. So they'll be like AT-AT, and then it'll repeat again, AT-AT, AT-AT, AT-AT. So they're short sequences that repeat, and they repeat many, many times. So here is a little short tandem repeat region. or a microsatellite region. Here the repeat is GATA, right? So every time you see a little yellow box, that's GATA, GATA, GATA. Now people can differ by how many times that repeats in their genome. So we're doing genotyping right here for the size of this microsatellite region, okay? This individual has 6 repeats. This individual has, or this chromosome has 10 repeats. So in microsatellite, these are known places in the genome, and so I can create primers that would allow me to amplify this region. So I put primers in the non-repetitive region that flank it, and then whatever size gets amplified is the genotype, right? So all we're talking about is what is the size of the fragment location here, okay? So if I amplify this, that's going to give me a 6 base pair piece. If I ran it on a gel, it's going to run quickly. This individual that gave this pattern, on both of their chromosomes, has 6 repeats at this microsatellite place. So they are a homozygous for the 6 repeat. This individual has the 10 repeat version on both of their chromosomes. So when you do the PCR, that you just get one unique band, same size on both chromosomes, so I just get a single band indicating that there was 10 repeats, because it's longer. This individual would be a heterozygote. One of their chromosomes has the 6 repeat, one has 10 repeat. Okay. There are 10,000 known microsatellite regions in the human genome. And so you can selectively go and make primers for 10,000 different regions and identify what the genotype of these individuals is for every single one of those regions. If you were to do that, you have generated what we call a DNA fingerprint, because you have a unique assortment of chromosomes. And so if we surveyed you for all of these microsatellites, you will have a unique imprint, a unique size at each one of those that's different than anybody else's combination. So we can use this for identification purposes. I went and grabbed a picture of a microsatellite gel online here. So this is our DNA standard, and then these are different individuals that we're sampling. And this is just at one microsatellite region. So this individual has 2 bands. This individual, they're faint, but there's one below and there's one a little bit above. This individual has these 2 pieces. This individual has a big piece here and a more common, smaller piece. So this is a genotype, right, for just this single location, one microsatellite and multiple different people. Now, some people, and I'm sorry that this one is light, but like these 2 people here, they basically have the same pieces. They've got a light band up here and a light band down here. Who else is identical? Well, I guess nobody else is identical. You might be identical with somebody else at one place, right? You might have a 6 base pair repeat and a 10 base pair repeat. Somebody else at that location might have a 6 and a 10. But if you go looking at all 10,000 or even just 1,000 of them, you're going to have a unique complement at these 1,000s that nobody else is going to share with you. Right? So this is actually the most common way that we identify people's DNA at crime scenes, for instance. If you get a sample of blood or tissue or something at a crime scene and you try and identify who that belongs to, this is what you would do. You would amplify, several thousand of these microsatellite regions on the sample that you found at the crime scene and all the people that you think are suspects, and try and find a match between the 2 of them. So this is DNA fingerprinting, is a type of genotyping. It's just what size pieces do you have at these variable regions. Yeah.
Were you saying that they can use this to screen for diseases or known diseases?
You could, if you knew that a certain diseased allele was really close to a known length microsatellite region, right?
Oh, okay.
Right. If the diseased cystic fibrosis allele sat right next to a 6 base pair repeat microsatellite, then I guess you could, right? Because if you have the 6 base pair repeat, then you probably have the mutant allele, right? If they were really closely linked. Usually microsatellites, they don't sit quite that conveniently to the genes that we're interested in. So you'd have to do it a different way. But this is just an example of genotyping, right, for fingerprinting purposes. Here is restriction mapping, and this example actually would be to allow you to identify a mutant allele. Here is the globin gene, the beta globin gene in hemoglobin. The normal beta globin gene contains a little sequence in here that is cut by the restriction enzyme DDE1. So it's a very short little, I think it's only a 6 base pair sequence. And a normal wild type functional beta globin gene has the sequence that gets recognized. So you take that normal piece of DNA and you cut it, and what you'll get is 106 or 175 base pairs here. It cuts in the middle, 201, and then it cuts over here. So a normal allele is going to give you these 2 fragments. So this individual, we took DNA from them, isolated the DNA from the rest of the cellular contents, digested it with this enzyme, and I'm getting only the 2 normal cuts. That says both of the chromosomes get cut in the normal manner, and so I get these 2 fragments. In the sickle cell anemia mutation, the mutation that causes this gene to be non-functional, the actual nucleotide changes are right there in that sequence where DDE1 recognizes. So in those 6 nucleotides, the sickle cell mutation changes one of those nucleotides. So if I were to soak this piece of DNA in with DDE1, it would cut out here, it would cut over here like the normal one, but the mutation that's causing the sickle cell disease makes it so that doesn't cut, right? So this is restriction mapping, or restriction genotyping. Because if an individual has this allele, if it's homozygous for that allele, it's only going to give this one big piece, right? So this is a sickle cell individual. It's got 2 mutant copies, and I have genotyped them. This would be a carrier. They've got 1 mutant copy, but the other chromosome carries the wild type. So here I could actually survey a population of people and see how common is the sickle cell mutant allele. I might have people that don't have sickle cell disease, but they actually do have that allele. That would show up in their genotype, right? They would be normal functioning people that wouldn't have sickle cell anemia, but by their genotype I could tell, well, they're carrying the mutant allele. They have a risk of passing the disease on to their child. Right?
Can they see this in utero, like when the baby's being developed?
Yeah, you can go and do an amniocentesis. So you go in, take some of the amniotic fluid out of the amniotic sac, and in that fluid is going to be some cells that the baby has just sloughed off during development. You can isolate DNA from those cells and do genotyping. So the whole industry of genetic counseling is based on that, like taking DNA from the embryo, from the amniotic fluid, doing genotyping on it and determining whether or not the baby actually has certain diseased genes or not, way before that disease would ever show up, right? Sickle cell anemia doesn't show up in utero because it's the mother's blood system that's doing all of the exchange of oxygen. It's when that baby's born and actually has to start breathing on their own that their phenotype would show up. So you can do pre-screening of this. You could also do pre-screening on in vitro fertilized embryos. In vitro, you can take an egg, you can take sperm, fertilize them together, and they'll start developing just in the petri dish. You grow them, typically what's done is people grow them to the stage that they normally would be implanted in the womb of the mother. At that stage, they can actually tolerate losing a couple of cells. So you can take that embryo that you've in vitro fertilized, pull off a couple of cells and do genotyping on them. And so you can determine which embryos have what traits at some level, right? And so then there's this whole, it's bordering on eugenics, well actually I think it is eugenics, of deciding which embryos to implant based on what genotype do they have. If you want a male, Well, you do a genotype and you just implant the embryos that have a Y chromosome based on your genetic analysis. Or if you have a risk for a disease, you might have diseased embryos that you've conceived by in vitro, but people will selectively just implant the non-diseased individuals and let the others die. But that's a lot of what goes on in genetic counseling right now, is screening for what traits you want to actually, which embryos you want to grow up based on their traits. This is ridiculously immoral, in my opinion. Just letting people die because you don't like the traits that they possess. But this is going on right now. You could do this if you want. This is you could also do DNA fingerprinting by restriction fragments. So here I've got a blood stain at a crime scene. This is DNA that that was isolated from a crime scene. So they took the blood sample, took the DNA out of it, and digested it with a restriction enzyme. And that's just going to fragment the genome and create these fragments. Then you can take all the suspects that you think were at the crime scene, take DNA from them, do the same fragmentation on them, and see which ones line up. You can see this individual represents the exact same banding pattern. So whoever this was lost some blood at the crime scene, right? You have to see what the other evidence is to say whether they were the victim or the perpetrator or an innocent bystander. I don't know. But this is just randomly fragmenting the genome, running them side by side. This is a form of genotyping, right? It's not specific to a disease, it's not specific to anything in particular. It's just what does the DNA look like when you fragment it. Right? So genotyping is a very generic form of looking at things. Now you can make it specific if you have specific regions, but in general it's a generic method.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.